about the DNA. Okay, deoxyribonucleic acids. What is the building block of nucleic acids? What is the building block of DNA? What is ribose sugar? Like that, we discuss all this in this DNA topic. Okay. First of all, what is the DNA? And what is the full form of the DNA? DNA is the deoxyribonucleic acid. What? Deoxyribonucleic acid. From name, it is clear that this is the deoxy. Means they don't have oxygen atom at the 2' in the ribose sugar. See, this is the ribose sugar, right? This is the ribose sugar. 1', 2', 3', 4', and here is 5, right? This is the ribose sugar. Each prime having OH group and HI. OH group, right? So, in DNA, these two prime don't having OH group. This is only hydrogen atom will be present here. So, this name is come from that, means there is deoxy, means there is no oxygen. So, this is the deoxyribonucleic acid. What? Nucleic acid. Means nitrogenous base these are acidic in nature. What? DNA is in acidic nature. From name it is clear that DNA is what nature? This is the acidic in nature. The chemical substance is present in the nucleus or cells in all living organisms. It is either eukaryotes or it is either prokaryotes, both having a DNA. Either this genetic DNA is actually called as the genetic material. What? Genetic material. Because DNA contains all the information of the cells. Here, but in this chapter, in this biochemistry, we discuss only about the biochemical nature of the DNA, not genetic nature. Okay. So, DNA are present in all eukaryotes or prokaryotes. In prokaryotes, DNA are flowed in cytoplasm, whereas in eukaryotes, they having a outer membrane that is called nuclear membrane. They are present in nuclear membrane, or they are present in the nucleus. DNA control all the chemical changes which take place in the cell, right? All the chemical changes, all the information that are encoded for protein or RNA, that all are controlled by this DNA molecule. That is deoxyribonucleic acids. The kind of cells which is formed is controlled by DNA. Means which type of cells will be formed in our body, like muscle cells will form, or blood cells, nerve cells. Which type of cells, which type of enzymes, which type of protein, that all are controlled by DNA. Because DNA code the sequence or information for all the formation of the cells. The kind of organism which is produced is controlled by DNA, like buttercup, giraffe, human, every kind of organism. Every kind of organism are controlled by DNA. Only and only the element that which control the whole organism, whole the structure of organism, that is the DNA. subunits are nucleotides, right? Subunits are nucleotides. Each nucleotide is made up of deoxyribose and a phosphate group and an organic base. See here. This is the structure of ribose, that is deoxyribose, right? Okay, this is the structure of deoxyribose. Deoxyribose, clear? A phosphate group are present here. Okay, this is the phosphate group are present here, and the organic base. Organic base are present here. This is the nitrogenous base. What? This is the nitrogenous base, that is maybe adenine, cytosine, guanine or thymine. Okay? This nitrogenous base are maybe adenine, cytosine, guanine or thymine. So this is the structure of the DNA. Not DNA, this is the molecule by which this chain of the DNA is formed. Now by this nitrogenous base are attached by phosphate group, another like this, and here a hydrogen bond will form. Okay, clear? So DNA is a very large molecule made up of a long chain of subunits, that is nucleotides. This is called as nucleotide. What? This whole structure is known as nucleotide. And this nucleotide is made up of phosphate group, deoxyribose sugar and organic base. Right? How chain will be formed? This phosphate group react with another this hydroxyl group of another ribose sugar, and like this polymer of chain will be formed. And this nitrogenous base will form hydrogen bond with in parallel of this another DNA molecule. Right? Clear? So ribose and deoxyribose. Ribose and deoxyribose. Ribose is a sugar like glucose. Ribose is nothing but is a sugar like glucose, but with only 5 carbon atom in its molecule. This is a 5 carbon atom. 1, 2, 3, 4, 5. Right? Deoxyribose is almost the same but lacks 1 oxygen atom. Deoxyribose what? It is nothing. Already I told it is almost the same but lacks what? 1 oxygen atom at 2' end. What? At 2' end. Both molecules may be represented by the symbol. This is a symbol. This is a 5 carbon atom, that is ribose, but purine and pyrimidine. What? Purine and pyrimidine. Purine and pyrimidine. This is purine ring and this is pyrimidine ring, right? Purine ring is made up of 2 chains and pyrimidine is having single chain. Okay. This is the pyrimidine ring, whereas purine ring having 2 ring. This is the purine ring. In pyrimidine, three nitrogenous bases are present. That is cytosine, uracil, thymine. Whereas purine, 2 nitrogenous bases are present. That is guanine and adenine. What? Guanine and adenine, right? This 3 type of pyrimidine. In this 3 type of pyrimidine, in DNA, two pyrimidine are present. That is cytosine and thymine. What? That is cytosine and thymine. Whereas in purine, both are present in DNA or RNA. In RNA molecule, uracil and cytosine are present. Uracil and cytosine are present. Okay. So uracil are not present in DNA and thymine are not present in RNA molecule. Instead of thymine, uracil are present in RNA. Instead of uracil, thymine are present in DNA. Whereas both guanine and adenine are present in both DNA and RNA molecule. Okay. Remember this structure. This structure is very important. Okay. Now, see here. First is this is the structure of DNA or ribose sugar in RNA. Okay. In DNA, what is the difference between DNA and RNA? These both structure are same. Only are different in structure. Only difference at this 2 primate. In DNA, it is having only H group, hydrogen atom in DNA. Okay. Whereas in RNA, here having OH group. In RNA, only difference is here, only and only. And another difference is in nitrogenous base. That is DNA having thymine and RNA having uracil. Okay. Due to this OH group that is present in RNA, make RNA more reactive as compared to DNA. So DNA is considered as the genetic material of the cell, because DNA is less reactive compared to RNA, because here only hydrogen atom is present that not react with another molecule, whereas this hydroxyl group react with another molecule, right? So DNA is less stable compared to RNA. Understand? It's clear? What is the difference between DNA and RNA? This is the sugar, that is ribose sugar. This ribose sugar having phosphate group, and here having nitrogenous base, that is organic molecule, and here is H atom or OH in RNA or H in DNA, and here it is OH, hydroxyl group, 3 primate, right? And this is the cytosine. This is the structure, how this link this cytosine structure, right? This cytosine, this is the carbon atom. This carbon react with this nitrogen atom, and ring of this nitrogen atom and form a bond. Like that only, all the nitrogenous base attached with this carbon molecule at one point, right? A, thymine is coded by T, cytosine is by C, and guanine is by G. And RNA, there is another, that is U, coded by U, uracil. Uracil is coded by U. Okay. These are the four nitrogenous bases or organic bases. This is nucleotide. What is nucleotide? When nitrogenous bases attached with ribose sugar, and that ribose sugar having also a phosphate group, then this all structure is called as nitrogenous base. What? Remember this. There are so many confusion. What is nitrogenous base? What is nucleotide? What is organic base? This all are more confusing. Okay? So clearly understand what is nitrogenous base. What is nucleotide? What is nucleotide? Understand what is nucleotide. Remember, if tide will be attached, means phosphate group is present. The deoxyribose, the phosphate and one of the bases combine to form a nucleotide. What? They all combine to form a nucleotide. Remember, phosphate group are present in this. What was present in nucleotide? A deoxyribose, phosphate, and one of the bases. One of the bases means from adenine, cytosine, thymine. From these all four, one organic bases are present. One organic bases are present in this, attached with this deoxyribose. Then that is the whole structure we call as nucleotide. This whole structure we call as nucleotide. Okay. And what is nitrogenous bases? If only adenine, guanine, cytosine, these are called nitrogenous bases. What is nucleosides? Another is, what is nucleoside? Nucleosides means this phosphate group are not attached. Only deoxyribose sugar and this adenine will attach, then this is called nucleoside. What? Nucleosides. And if this all are attached, then it is called nucleotide. Okay? Clear? What is nucleotide and what is nucleoside? Now, next is how these nucleotides are joined. A molecule of DNA is formed by millions of nucleotides that join together in a long chain. Like you already know that how the protein will join with each other. Protein is a long chain of amino acids. Like that only, DNA is the polymer of nucleotide. What? DNA is the polymer of nucleotide. They are joined in a long chain. See here, this all are what? This all are nucleotide. This single chain are what? These are nucleotide. This nucleotide joined by phosphate and OH group of second prime. Means this is the 5' end and this is the second prime end. This is the 5' end and this is the second prime end. So this is the 3' end. This is the 3' end. So always bond formation will be takes place at the the 3 prime of the OH group of the nucleotide. Okay, and form a bond. What? Form a bond, that is called phosphodiester bond. What? Phosphodiester bond. This bond is called form a bond, that is phosphodiester bond. And this bond, after this bond will be formed in between, and this like that only this bond will be formed, and a long chain of nucleotide will be arranged. And this long chain of the nucleotide is the sequence of the DNA. What? This is called as a polymer of the nucleotide, and this is a DNA structure. And this is here is a base A, T, C, G. One of these base will be present here. Either A, either T, either G. One from this all. Okay. So polymer will be formed at the. So what happened? If here if here phosphate group is present, then it is known as 5 prime end, and if here OH group is present, then it is known as 3 prime end. So DNA having end, that is a 5 prime end and a 3 prime end. What? 5 prime end and 3 prime end. This is called as 5 prime end and 3 prime end. Like in protein there is a N terminal and C terminal. Here in DNA there is a 5 prime and 3 prime. If hydroxyl group are present in last, that is the 3 prime end, and if phosphate group are present in last, that is known as 5 prime end. So remember this. Where is the 5 prime end will become, and where 3 prime end will become. This is a sugar phosphate. This is called as sugar phosphate backbone. What is this? This is a sugar phosphate backbone. Sugar phosphate means this sugar and phosphate will be occur, and this is a back. So this is known as backbone. Backbone. In fact, the DNA usually consists of double strand of nucleotide. Generally, DNA is of double stranded, like this. Like this. Okay. Sorry. usually consists of a double stranded of nucleotide. The sugar phosphate chain are on the outside and the strand are held together by chemical bond between the bases. Okay, and bonds are between the bases. What type of bond are present in between the bases? That is A, T, G, C. What type of bond are present in these two? The bond present in this nitrogenous bases or organic bases are the hydrogen bond. What? Hydrogen bond. If A will, another thing is what? Always purine will combine with pyrimidine. Okay, pyrimidine. Purine will combine with pyrimidine. Okay, what happened? Purine combined with pyrimidine. Purine combined with pyrimidine, and there are specific bases which combine with specific base of purine is combined with a specific base of pyrimidine. Not all purine bases will combine with all pyrimidine bases. Means any of the purine bases combined with any of the pyrimidine bases, this is not occur. Means adenine combined with thymine. Adenine combined with thymine. Guanine combined with cytosine. Okay, and adenine when combined with thymine, they form double hydrogen bond, and when guanine combined with cytosine, they form triple hydrogen bond. So guanine and cytosine are more stable compared to adenine and thymine. Okay, so this line is clear now. The sugar phosphate chain are on the outside, that is sugar phosphate chain are present as a backbone. What? They are present as a backbone of the DNA. And these strands are held together by a chemical bond that are between the bases, means that are by the hydrogen. That are by the hydrogen bond that are present in between the bases, that is AT or CG. Okay, clear it is? See, these are the two-strand DNA. This is the first strand of the DNA, and how the other strand will come and combine with this strand. If there is A, then here T thymine bases will come and form bond with A. Not G base will come and form with adenine. No, never. Only thymine will come and bind with adenine. Okay. If their G will be present, then your cytosine will come and form 3 hydrogen bond with guanine. Okay. And if here cytosine is present, then here guanine will come. Guanine base will become nitrogenous base will come and combine this. And if here thymine will be present, then here adenine will become and combine with 2 hydrogen bonds. Okay. Remember Okay. See here, how the bonding will be formed. The bases always pair in the same way. Adenine form a bond with thymine. Form adenine bond thymine, that is by 2 hydrogen bond, and cytosine bond with guanine. Already I told you, by 3 hydrogen bond. Okay, understand this slide is clear. Now this is how bond formation will takes place. Bonding 2, how bond formation will takes place. Same it is. This is one strand. This is the one strand of the DNA. This is one strand of the DNA. This is the second strand of the DNA. Here it is 5 prime end. Here it is 3 prime end. Here also there is 5 prime end. Here also 3 prime end. Okay, means they are parallel in nature. They are parallel in nature, huh? And both bases, nitrogenous base will came and combined with their complementary bases, and bond formation will be takes place. Okay. Now the pair strand are coiled into a spiral called a double helix. How the double helix DNA will be formed? Actually, when this nitrogenous bases will be formed, then there is helical formation will be takes place. Like alpha helix, you understand in a protein structure, like that only. There is a helical form will be takes place. That form, because hydrogen bond formation will takes place. During the formation of this hydrogen bond, there is a helical pattern of the DNA will be takes place. This is known as double helix DNA. What? This is known as double helix DNA. This is a sugar phosphate backbone. This is a backbone, and this is a nitrogenous basis by which DNA will be formed. Okay. This sugar phosphate chain provide support or stability to the DNA. Provide stability to the DNA. This sugar phosphate bond. Okay. Now this replication topic we cover in a genetics. Briefly, I told you what is replication. Replication is a type of division, type of increase the number of itself. Actually, it is the itself copy. Increase the number of itself copy. Before a cell division, the DNA strand unwind and separate. Each strand make a new partner by adding an appropriate nucleotide. The result is that there are now 2 double-stranded DNA molecules in the nucleus. So that when the cell divides, each nucleus contains identical copy. What is replication? Actually, what is replication? Nothing but it is a copy. Increase the copy number. Or it is the increase the duplicacy or duplicate their number. Okay. In genetics, we study more about this replication topic. Actually, we discussed about one type of question generally arise. Which of the following repeats a correct pairing of bases? Because we already in the starting we discussed that types of base, adenine combined with thymine, guanine combined with cytosine, that only. Okay. So what happened here? Which are the correct bases? Adenine combined with thymine, right? This is correct. This is correct. Adenine combined with thymine, and adenine combined with guanine? No. Thymine combined with adenine? No. Guanine combined with cytosine? Right. Thymine combined with thymine? No. So adenine combined with thymine and guanine combined with